diagnosed with pancreatic cancer. AGT established this lectureship to honor Gordy for his longtime contributions to AGT and to the cytogenetics community. Gordy started his cytogenetic career as a bench technologist at the University of North Dakota. While at UND, he received his PhD in cytogenetics. In 1972, Gordy moved to Rochester, Minnesota to work at the Mayo Clinic. From 1972 until his death, Gordy was recognized by cytogeneticists worldwide as a leader and innovator. His more than 270 papers cover virtually every area of cytogenetics, automation, quality, congenital disorders, oncology, and fish. And many of those papers set the standards for this field at that time. Gordy was a requested guest lecturer throughout the world, and he often spoke at AGT's annual meeting. Gordy supported our organization in many ways, but the most noteworthy may well be his assistance in getting us a role on the CAP Cytogenetics Subcommittee, the only technologist organization so recognized on that committee. Gordy was awarded the AGT Outstanding Achievement Award in 2002. The Gordon W. DeWald Lecture is a tribute to one of our outstanding colleagues who shall be missed and continues to be missed by those who knew him. This year's Gordon DeWald Lecture will be given by Dr. David Ledbetter. David is the Executive Vice President and Chief Scientific Officer at Geisinger Health System. Dr. Ledbetter came to Geisinger from Emory University School of Medicine, not too far away here, where he was the Robert W. Woodruff Professor and Director of the Division of Medical Genetics in the Department of Human Genetics. David previously held academic and leadership positions at the University of Chicago, the National Center for Human Genome Research at the National Institutes of Health, and Baylor College of Medicine. He is a graduate of Tulane University and earned his doctorate at the University of Texas in Austin. After his early discovery of the genetic cause of Prader-Willi syndrome and Miller-Dieker syndrome, Dr. Ledbetter has focused his research efforts on discovering the underlying etiology of childhood development, disabilities such as autism, and the translation of new genomics technologies into clinically useful genetic tests for early diagnosis and intervention. David's current research interest includes leveraging the massive amounts of genomics data generated during routine patient care for knowledge generation and integration of this information into electronic health records in a clinically useful manner. Let's give a warm AGT applause for Dr. Ledbetter. We have a plaque to present.
Thank you very much. It's a great honor to participate in honoring Gordy DeWald, who was a great friend and mentor to me and so many other people in the cytogenetics field. And we lost him much too young in his continuing productive and influential career. But thank you again for the opportunity. So, which is the advance here. Okay. First, I should comment about my consulting activities. I'm a consultant and member of Scientific Advisory Board for Roche Nimblegen, CombiMatrix, and Cellula. I also have a scientific disclosure to make. That is that copy number variation analysis, or CNV analysis, is really just a rebranding marketing strategy for cytogenetics. It's a study of deletions and duplications with a new name to get grant funding, get your papers published in a higher profile journal because it sounds new and exciting and molecular rather than conventional cytogenetics. My clinical cytogenetics career started and was stimulated by this young woman, Caroline, who has Prader-Willi syndrome. I was originally an evolutionary biologist and headed on a basic science career studying primate chromosome evolution. During my part-time job as a technician in the cytogenetics lab at Baylor while I was a graduate student, we discovered the deletion of chromosome 15, and Caroline was the first patient that deletion was observed in. So that moved me into the human clinical cytogenetics arena away from my basic science early training. I was hired in 1981 as the director of the Kleberg Cytogenetics Lab at Baylor by Tom Caskey, who was chairman of the department and a distinguished human geneticist and early pioneer in the application of molecular genetics. But when Tom Caskey hired me as the director of the cytogenetics lab, his first advice to me was to throw away my microscope because cytogenetics would become extinct within the next 5 years. Fortunately, this prediction was not correct, as evidenced by the extent that cytogenetics is still used both in research and clinical applications. Although we've added a lot of molecular technology on top of cytogenetics. And looking forward to the future, there will be an increasing role of molecular versions of cytogenetics. So it's interesting to think why the human G-banded karyotype has been so powerful and has remained a primary research and clinical tool in the face of this onslaught of new molecular technology starting in the 1980s when Tom Caskey and others predicted that southern blotting and RFLPs would be the way that we diagnose Down syndrome and other chromosome abnormalities. And the way I view it is that cytogenetics was the first whole genome analysis. We could examine and analyze the entire genome in a single microscopic field. And to replicate that with molecular techniques, you needed at least 500 or 600 perfectly spaced DNA probes and a technology that would accurately detect single copy variation as well as karyotype analysis did. So we've been doing CNV analysis of the whole genome since 1956. So one question I get asked a lot by cytogeneticists, including cytogenetics technologists, is will cytogenetics survive in the future, or how long will cytogenetics survive? And to this I go back to an evolutionary biology framework of thinking and quote the famous evolutionary biologist Winston Churchill, change is the price for survival. So if you're willing to change and adapt, the future is promising. For those uninterested in change and adapting, then the future may not be quite as optimistic. So it's interesting. It's been interesting to me to watch the new generation of genome scientists rediscover the phenomenon of gene dosage, which of course we've learned a lot about gene doses from karyotype analysis over the last 50 years, and all cytogeneticists are familiar with the fact that a visible deletion or duplication anywhere in the karyotype causes intellectual disability, previously known as mental retardation. And that there are polymorphic regions in our genome or in our karyotype in normal individuals, particularly in the pericentromeric regions, acrocentric short arms, the Y chromosome in males. So the notion of copy number variation that has a benign impact on individuals versus copy number variation that has a pathogenic effect is quite familiar to cytogeneticists. It's not a new phenomenon. It's only a new phenomenon to a new generation without adequate cytogenetic training. The other lessons we've learned from cytogenetics, and it's, again, amazing to me, my molecular biology and genomics friends are astounded that cytogeneticists know these facts. One is that monosomy or deletion is worse than trisomy or duplication of the same size chromosome region. The second one is the one that I really had a hard time keeping a straight face when my colleagues told me that there was no evidence to support this, is that larger imbalances, therefore involving more genes, are worse than smaller imbalances. Sounds pretty straightforward to me. And we now have molecular evidence to prove this is true, in case you were waiting for that. We've known for a long time that imbalance of gene-negative bands, which are gene-rich regions, is worse than imbalance of G-positive bands, and the Human Genome Project has proven that to be true. Not all genes are dosage sensitive. We know that from Down syndrome, where partial trisomy of the distal terminal region of chromosome 21 gives most, if not all, of the features of Down syndrome. So the rest of the chromosome is not contributing. So you can infer from that that there are other genes that are not dosage sensitive, so an extra copy is not contributing to the abnormal phenotype. Maybe the best example is Angelman syndrome, where point mutation of a single gene, UBE3A, gives the same phenotype of a 4 to 5 megabase deletion, including about 30 genes. That tells us that the other 29-ish genes in that region aren't contributing in a significant way to the phenotype. Only one is the major culprit for the Angelman syndrome phenotype. So when I lecture about gene dosage and try to explain when CNVs were first rediscovered in normal individuals, everybody was shocked that normal people could have deletions or duplications. When you think about it, it's not so surprising, and because we don't really know how many genes in our genome are dosage sensitive and require exactly 2 copies for normal development and normal function. In fact, most people would guess that it's a minority of genes that are dosage sensitive, and most genes having 1 good copy is enough. Autosomal recessive genetic disease is a good example where mutation and activation of 1 gene doesn't cause an abnormal phenotype. So in this cartoon, I've shown a string of 10-ish genes. The open circles are not dosage sensitive, so you only need 1 copy to have full gene expression and normal development. The half-filled or half-blackened circles are dosage-sensitive genes, and you need 2 copies to have enough gene product for normal development. So if you have deletions, shown by the bars above the chromosome there, small microdeletions or small CNVs that only knock out genes that are not dosage sensitive, there's no phenotype involved. So you can have lots of CNVs in normal individuals until you get to a gene that is dosage sensitive. And if that's only 1 out of 10 genes, you need a big deletion. So a visible deletion would knock out 50 genes on average. So that would be big enough to knock out several dosage sensitive genes and produce an abnormal phenotype. But it actually, in retrospect, makes a lot of sense that small deletions with either no genes or a small number of genes that are not dosage sensitive are tolerated in our genome without any significant selection against that individual. So you're all familiar with various formats for microarray, so I'm not going to talk too much about the technology. One of the popular methods is comparative genome hybridization, but all of the methods, SNP platforms, and as well as ArrayCGH, are now very sensitive to copy number changes, single copy number changes. So very accurate at identifying deletions, duplications, and now even mosaic deletions and duplications. And as you're aware, reviews of the literature, including this one from our ISCA consortium group published in 2010, reviewed the literature and showed that the frequency of clinically significant CNVs in children with unexplained intellectual disability, birth defects, autism is about 15 to 20%. And this may impact prognosis and in many cases now direct management of comorbidities, other medical problems that can be anticipated in children once you identify a pathogenic copy number variant. So the American College of Medical Genetics in September of 2010 made this statement recommending chromosome microarray as the first tier test in all children with unexplained developmental disabilities, including autism. I'm surprised there's still some physicians ordering G-banded karyotype first, and labs should be educating those physicians. They should be doing array first, and once array is done, there are very few situations where a karyotype adds any additional value. But we've learned over the years that you have to think about the array design, and from the beginning, our consortium of clinical cytogenetics labs focused on designing arrays that were not the highest resolution research arrays that covered the entire genome, but only had a resolution and coverage that was useful in a clinical diagnostic application. So I refer to this as a clinical array design as opposed to a high-resolution research array. Most array designs now cover the entire genome, although we usually exclude the repetitive region of the genome because we don't want to see all of the normal benign CNVs that occur in every individual. So we'd rather not deal with 5 or 10 or 20 CNVs in every normal individual and every patient. Instead, we focus on the unique part of the genome that contains genes that have clinical relevance. But you can alter the resolution as shown in this cartoon first published in 2008 in Genetics in Medicine from our group at Emory at the time, and now has been used to inform the ISCA recommendations and are similar to the American College of Medical Genetics recommendations on array design. And most everybody in the audience now is familiar with actual analysis of these arrays. In this example I show on the left, a typical 4 to 5 megabase deletion in the Prader-Willi-Angelman syndrome region. And I will comment that looking at these is not nearly as satisfying as the beautiful aesthetics of looking at G-banded chromosomes. So it's sad that we don't get to look at chromosomes. However, we do get a higher yield and are able to offer better clinical diagnostic testing for our patients. So, on the right is a much smaller deletion, and if I blow that up, it shows a small deletion of only 45 kilobases, which is only part of the UBE3A gene. This patient had clinical features of Angelman syndrome and had had FISH testing, which was normal, had had methylation testing, which was normal, and had sequencing for the UBE3A gene done, which was normal. So the physician then ordered array and found a deletion in the UBE3A gene that was missed by sequencing because you sequence the normal allele on the other chromosome and you can't detect copy number variation by current clinical sequencing technology. That may change in the future, but at the moment, we don't typically identify copy number changes. So in this case, array was the only method able to detect this mutation, and there was no other molecular diagnostic method capable of doing it. More typical examples of what we find in clinical application, children with unexplained non-syndromic developmental abnormalities are deletions of several megabases, as shown here. 2 unrelated patients, both with deletions on the long arm of chromosome 7. The beauty of array analysis is once you identify the deletion, you can immediately take the genome coordinates, go to the genome browser, and see what genes are involved in the deletion. And when you do this, and plot the 9 megabase deletion of case 3 and 2.5 megabase deletion in case 4, you get a display of all of the genes that are deleted in each of these. And if you look at the common region of overlap, there are 2 known genes that are co-deleted that correspond to Mendelian genetic disorders known to be autosomal dominant loss of function. So we know that these patients have risk for these disorders, and particularly the one on the top is a dominant long QT or cardiac arrhythmia syndrome, and these children and individuals are at very high risk of sudden death due to cardiac arrest on the playground, playing sports, etc. So this child there was no positive family history, there was no reason to suspect any cardiac abnormality or arrhythmia, but based on the chromosome microarray done for developmental delay, identified a critical medical risk and was confirmed by EKG just prior to a surgery this child was scheduled to have. So sometimes there can be life-saving information that's identified through this kind of whole genome analysis. Another case, child again, developmental delay dysmorphic features with a deletion on chromosome 17 to a little over 2 megabases. When you look at the genes that are deleted, it includes the famous tumor suppressor gene P53. So this child has Li-Fraumeni syndrome and very high risk of a number of different cancers with no positive family history, no reason to suspect any increased cancer risk other than this clinical cytogenetic result, which led them to refer the child for a tumor monitoring protocol to try to detect early the onset of any cancers. So this leads to not only an increased yield for chromosome microarray clinical testing, but we often find what some people call incidental findings. The college is now using the term secondary findings that can lead to life-saving interventions, critical medical management decisions, although as a field, we've done a pretty poor job to date in systematically collecting data on the clinical utility and value of all genetic testing, but chromosome microarrays included in that group. We see the benefit immediately in the lab and in our genetics programs, but we haven't documented and collected evidence in a systematic way to convince insurance companies and other non-genetics healthcare providers. There's sort of an irony about the results of chromosome microarray to me, and that is children with developmental disabilities who in general I would call a relatively disadvantaged group that doesn't always have all of the appropriate medical care and then following social services and et cetera, are one of the first beneficiaries of the Human Genome Project and truly predictive, personalized medicine. So I think the application of chromosome microarray is one of the first home runs from the Human Genome Project, and moving that technology rapidly from the research lab into the clinical lab to improve diagnostic yield, but then the quality of the information that comes from that has much greater clinical utility than previous methods by chromosome analysis only. The next challenge is how do we put all this data into electronic health record or patient's personal health vaults in a clinically useful way to inform their physicians and other healthcare providers? And then how do we update that? Because currently the average deletion or duplication we see is 3 or 4 megabases, has 50 or 100 or several hundred genes involved, and we only know the clinical significance of 1 or 2 of those genes in an average case. But next year, somebody's going to publish the clinical disease association of one of the other genes that you found in a deletion last year. How do we go back and update our clinical interpretation as knowledge rapidly increases on the clinical significance of the many genes that are deleted or duplicated in a typical microdeletion. So my interest over the last few years is that now when we do routine clinical testing with this powerful new technology on tens, probably hundreds of thousands of patients now in the US are getting whole genome microarray analysis. This is increasingly being better reimbursed by insurance companies. So there's more CNV data generated during routine patient care in clinical labs than in all the research labs in the world. So, and that data is free, since it's paid for by insurance. So the challenge is how do we collect that data and clinical phenotypic data in a way that we can do data mining to improve our knowledge and provide even better information and better patient care in the future. The same is going to be true for DNA sequencing data, where we're starting to do whole exome sequencing on a clinical basis. If we could capture all that data in a single public database somewhere, then it would be a powerful research discovery tool, and it would accelerate our being able to provide much more information to our patients. So with this in mind, Krista Martin, who spoke yesterday afternoon, and I at Emory started a small informal group that evolved quickly into an international consortium around cytogenomic array data initially, and now is moving into sequencing data. And the notion was getting clinical labs to submit all of their data to a central public database, so any researcher could do data mining to do genotype-phenotype correlations, and then that new knowledge would improve our ability to interpret clinical microarrays and then sequencing in the future. There are over 150 labs internationally now that are members of this organization. The first analysis we did of the data was when we had about 16,000 cases in the database and compared CNVs to over 10,000 published controls. In that process, we were able to use typical GWAS-type statistical analysis to document new pathogenic CNVs. Some of these were CNVs that different labs were interpreting or guessing whether they were benign, pathogenic, or uncertain clinical significance. So this allowed us to do a formal statistical analysis. And from that, for example, the deletion 16p11.2, which was initially classified as a variant of uncertain clinical significance, now could confidently be called a pathogenic CNV with very high statistical confidence. Another study led by Evan Eichler using data from Signature Genomics and a similar approach actually identified 59 pathogenic CNVs. So now from these two large studies, we've taken the known pathogenic CNVs, put them in the ISCA database, so you can review that, and it also has a track on the UCSC Genome Browser, so when you're analyzing your own cases, you can consult ISCA or UCSC Genome Browser and see what the known pathogenic as well as known benign CNVs, so you don't have to struggle with interpretation when there's sufficient statistical evidence from very large population studies. On the left is a list of the labs that are contributing data to the ISCA database. If your lab is not there, shame on you. Go back and talk to your lab director and correct that. And then a lot of international labs outside the US as well. We did a survey of the members of ISCA recently, and you'll see that a third of the respondents to this survey were the laboratory cytogenetics technologists, shown in the blue piece of the pie here. The survey contained some simple questions. Does your clinical lab perform microarray testing? And 80% said yes. Does your laboratory submit data to the ISC consortium? Unfortunately, only a little over 20% said yes. So what are the reasons people are not contributing and sharing their data to improve our knowledge and improve quality of care to everybody's patients. And not surprisingly, it's lack of time, lack of resources are the primary thing. So our goal is to make this as easy as possible. And so on our ISCA website, there's a lot of information about how to prepare your data and submit your data. But in addition to that, we've had a goal for a long time of making it so easy for the clinical lab that at the end of every day or the end of every week, you click one button and your data automatically gets submitted to this database so that it doesn't interfere with your busy workflow and throughput. And we've recruited our industry partners, the Array CGH software and array providers, to help us with this. And all of the companies shown here are in various stages of developing software tools to allow you this one-click data submission option so that it doesn't take you any time and we get rid of that excuse for not contributing your data to this database. Now, there's another aspect of data submission that I didn't fully appreciate when we started this. Krista Martin has recently spoken on this topic at our ISCA conference a few weeks ago in Bethesda, Maryland, and that is the value of sharing your data and then comparing your data to other people's in this central repository. So you're all familiar now with the 5 categories of classification of CNVs by the American College of Medical Genetics, led by Hutton Kearney and others. Now on the ISCA database project, when you submit your data, we have a known ISCA pathogenic deletion track or categorization. So we have a designated known pathogenic, known benign category. And if you submit something that has the same breakpoints, the same deletion or duplication as on this track, but you try to call it something else, so you classify it differently from what we've declared as the known pathogenic, it flags it and sends you a little message to inform you this is a known pathogenic. You called it a variant of uncertain significance. Would you like to reconsider your classification? So you get rapid feedback. What was interesting when we started doing this from Emory, we had 3 different lab directors signing out cases. We all thought we were using the same rule set, but when we submitted our data, we got back a message saying, Emory has submitted 2 different cases with exactly the same breakpoints, but you classified them differently. And I was shocked at how often that happened. And then if you submit data from Emory, for example, and there's data in the database from 10 other labs, it'll send you a message saying 5 other labs saw this same CNV. They called it pathogenic, you called it benign. Would you like to reconsider your classification? So as I said, Krista has done an analysis on this, and in fact, the average conflict frequency is about 5%. I was shocked. So now, to me, if you're not participating in this quality control program, you are not providing the highest quality care for your patients. So when I think about what do I need to do to motivate people to contribute their data to this database project, I started out thinking, well, we're all pretty altruistic and we want to contribute to research. And we know that that research is intended to improve our knowledge and improve patient care. Sometimes there are database projects like this where you promise, okay, if you submit enough data, you'll be a co-author on all the papers that come out. So academic credit might be a motivator. One of the reviewers of one of our publications from this project raised the interesting point that it would be almost unethical with a new technology and at a critical point in understanding how to interpret this to not share your data. So this is the guilt category. But now I think the most important, clearly the most important, is quality control. And as I said, I didn't anticipate this to be as important it is. But I think it's so important now, I could see labs marketing that we're not only CAPA approved and CLIA approved, but we participate in the ISCA quality assurance program, and my competitor doesn't. Why would you send your samples to a lab that doesn't participate in the most important, most valuable quality control program available to cytogeneticists? Now, with this new technology, obviously we find a lot of new deletions and duplications that we weren't familiar with by standard karyotype, and so we have struggled with determining what's pathogenic, what's benign. The ideal is when you look at evidence from the literature and from databases like OMIM and Decipher and DBVAR at NCBI, Database of Genomic Variants in Toronto, etc., you find other cases with the same CNV and the same phenotype as your patient, so it's pretty straightforward to interpret either as a CNV in the normal population, so it's benign, or CNV in multiple patients with a similar phenotype. Unfortunately, we find a lot of very rare or even private family mutations, and then we're left to use our clinical judgment based on the size of the CNV, whether it's a deletion or duplication, how many genes and what genes are present in the CNV, and then to look at family studies and look at the parents to see if it's a de novo event or an inherited event, similar to the way we've interpreted novel karyotype findings throughout cytogenetic history. But when you have this clinical judgment, as we've already alluded to, you have significant variability. Some labs and lab directors are more conservative, some are less conservative in their interpretation. And the solution to this is to eliminate clinical judgment as much as possible by having statistical analysis that proves which are pathogenic, which are benign. So we need a whole lot more data in our database. But part of the misconception and confusion around these new, smaller microdeletions, or CNVs, has to do with the fact that there's significant phenotypic variability. Again, something that cytogeneticists and clinical geneticists are used to, because virtually all chromosome disorders, including Down syndrome and other classic aneuploidies, are very variable in their clinical presentation and which features are present. So this is not new. It just creates some confusion and anxiety when you're dealing with a new microdeletion and we don't have enough data on the range of phenotype. And geneticists always invoke variable expressivity and incomplete penetrance, but they rarely tell you which one they're talking about and make clear which one they think is most important for any particular genetic mutation. Now, variable expressivity means you can have a range of clinical manifestation from very mild to very severe, but everybody or most people have some phenotypic expression. Incomplete penetrance means some people with a genetic mutation, in this case CNV, appear to be normal. They appear unaffected. They appear normal. But of course, penetrance and expressivity depends on which phenotype and what kind of phenotype. As I'll illustrate here using IQ. With all of the problems of IQ testing, it's still a useful example to demonstrate this concept. IQ tests are designed to have a mean of 100 and a standard deviation of 15. And in medicine and for educational purposes, we need to have a way to identify who has intellectual disability. How do we do that? We somewhat arbitrarily create a cutoff point at 2 standard deviations below the mean, or an IQ of 70, and call anything below that cutoff intellectual disability. Anything above that we call normal. Now, for Down syndrome and Fragile X, that doesn't create any problems because everybody with trisomy 21 or Fragile X full mutation, the IQ distribution is shifted so far to the left, virtually everybody falls below the cutoff and is described as having intellectual disability, moderate to severe. But you see at the right end of that distribution, there are a few individuals that are called high-functioning Down syndrome or Fragile X, and their formal IQ scores may actually overlap into the normal range, but just barely. Now, if you take a CNV, and this is the most famous and well-documented CNV, the microdeletion of 22q11.2 associated with DeGeorge, VCF, and other phenotypic features, the mean IQ is 70 and the standard deviation is 15. So in fact, many individuals overlap the normal end of the distribution. And laboratory people know this because when you have a child and you identify the deletion, you ask for the parents. About 10% of the time, one of the parents has the same deletion. You call the referring physician, say one of the parents has the deletion, and before you can continue the sentence, the physician interrupts and says, I know which parent has it, because they met the parent. They're the ones who struggled in school, had a lot of learning difficulties, didn't match the educational, professional standards of the rest of the family that parent, that quote, normal parent came from. So, and that makes sense now because a lot of individuals with IQs in the 80s, for example, are walking around in the normal population and have children. But you can see by looking at cognitive performance as a continuous quantitative trait that this genetic mutation shifts the distribution to the left. So it's not incomplete penetrance that we have in 22q, it's variable expressivity, probably due to the rest of the genetic background or family background in that particular family. So, and the cost of this genetic mutation is 2 standard deviations at a population level. Similarly, when the deletion of 16p11.2, a new CNV came out associated with autism, there was quite a bit of controversy whether this really was pathogenic or not, because there were a lot of individuals with this deletion who were in the, quote, normal population. They were not identified as patients, but were parents of children who had autism or other developmental disabilities. And it turns out this deletion 16p11.2 is the second most common deletion we see in a clinical lab setting. So 22q11 is the most common, and 16p11.2 is the second most common. So we now have a lot of data, and in fact, we identify 2 or 3 such deletions every week through the ISCA consortium. Early on, there was an estimated risk of autism of about 50% when you have this deletion. The Simons Foundation in New York, the largest private funder for autism research, has started a project to collect 100 families with deletion 16p11.2 to do more detailed phenotyping, including brain imaging and IQ testing and autism diagnostic testing. If you look at the IQ results, it's exactly the same as 22q11.2. Now in this study, we actually have the parents' IQ shown in blue, and the parents who participated in the study actually had a little bit above average mean of 108. Their children with deletion 16p11.2 had a mean of 80, so the difference between the child with the deletion and their parents was 2 standard deviations. So this genetic mutation, deletion 16p11.2, costs 2 standard deviation from what the predicted IQ cognitive performance of the child would have been based on the family background. The same is true for social behavior. In this slide, I show SRS, or Social Responsiveness Scale, which is a quantitative measure that shows that in this measure, to the right, higher scores represent social impairment. To the left, low scores represent high social competence. In this case, the parents and the normal siblings had means of about 30, which is actually a little better than the population mean. So again, better than average for the families participating in the study. But their children with the deletion had poorer scores by almost 3 standard deviations. Now, there's still some controversy about the relationship of this deletion to autism, because only about 15% of these patients meet full diagnostic criteria for autism, but the ones who don't meet full criteria are very close. So they miss that cutoff, but they have a lot of social communication problems and behavioral problems. So the point of all this is to clarify that we need to be aware whether the phenotype we care about is a quantitative trait or is truly a qualitative or dichotomous trait. And now we have the ability to study families and to get phenotypic information on parents, typically developing siblings as well as probands with these CNVs. So to summarize this, it's my interpretation at least that we have good data now for 22q11 and 16p11 that penetrance is very high, perhaps even 100% on these quantitative traits, but there's variable expressivity and it may be dependent on the family genetic background and other environmental traits. And for 16p11.2, this carries over not just to cognitive performance, but to social behavior. So what about prenatal testing for chromosome microarray? Many of you are aware there's a multi-center study that's now completed data collection. There's a manuscript under review that we hope will be accepted and published in the next couple of months. This multi-center study is led by Ron Wapner at Columbia and involves microarray testing labs at Baylor, Columbia, Emory, and Signature Genomics. We studied over 4,000 patients referred for prenatal testing by Amnio or CVS, about evenly divided between those two, that were either routine AMA indications, ultrasound anomalies, or positive screening with elevated risk. And here are the key results from the study. The ultrasound abnormality category had the highest clinically relevant CNV, so pathogenic CNVs not detected by karyotype. So this 6% abnormal rate is on top of identifying aneuploidy and visible chromosome abnormalities. So very high clinical yield. But maybe what was more surprising than we all expected to see a pretty high yield for ultrasound abnormal pregnancies is that the AMA and positive biochemical screen pregnancies showed almost 2% clinically significant CNVs not detected by karyotype. So that led our group to these conclusions that direct uncultured amniotic fluid, or CVS, can be used for chromosome microarray analysis, so you don't have to wait for culture. Chromosome microarray reliably detects all of the aneuploidies and visible chromosome abnormalities identified by G-banding, but also detects another 2.5% clinically significant pathogenic CNVs not detected by karyotype. You break that 2.5% overall down to 6% when it's an abnormal ultrasound and 1.7% when it's a routine AMA or biochemical screen. So the conclusion and recommendation is chromosome microarray should be transitioned to the first tier prenatal diagnostic test in place of G-banded karyotype analysis. So I expect to see very rapid change and stopping G-banded karyotype analysis in prenatal diagnosis over the next year or so. What about non-invasive prenatal diagnosis? You're hearing a lot about that, both scientifically and from clinical service companies. The current non-invasive prenatal diagnostic clinical services are primarily able to detect trisomy 21 and other whole chromosome aneuploidies. But on a research level, people are using whole genome sequencing from non-invasive methods to identify single gene mutations. So the feasibility has been documented, but we need more work to determine accuracy and cost. It has not yet been able to identify smaller CNV disorders, and I've just shown you that those are very common even in routine pregnancies, more common than we expected and more common than Down syndrome. So for now, I would say CVS or amniochromosome microarray testing is the most sensitive prenatal test available, but of course, all of this technology is changing very rapidly. I'm running out of time, so I'm not going to be able to say too much about whole genome and exome sequencing, so I'm going to skip through this and say that exome sequencing is now being implemented both research and clinical applications, and there's evidence in autism and intellectual disabilities that you identify an additional 10 to 20% causative mutations. So for now, chromosome microarray would be considered the first-tier test, but clinical exome sequencing might be considered an appropriate clinical test as a second-tier test after a normal microarray. So if you fail to reach a diagnosis by microarray, it's appropriate to consider clinical exome sequencing. So my conclusions for postnatal cytogenetics, whole genome chromosome microarray is the standard of care. For intellectual disabilities and autism with yields of 20% and 10% respectively, and exome sequencing is becoming the second-tier test. For prenatal diagnosis, as soon as the results of the NICHD study are published, I expect the relevant professional societies will update their guidelines and recommendations around chromosome microarray. And non-invasive prenatal diagnosis, which although it's popular because it's non-invasive, I think we'll need to reliably detect small CNVs to be able to compete with the power and diagnostic yield of invasive chromosome microarray testing. Now, cancer genetics, and I'm sorry that I didn't do justice to the Gordy DeWald lecture in addressing cancer genetics, but it's not my particular area of expertise. As you know better than I do, cytogenetics is still critical to identify the many diagnostic and therapy-directing translocations in a variety of leukemias, lymphomas. And until we get exome genome sequencing technology more reliable and much cheaper than it is today, cytogenetics, G-banding, and FISH will continue to be the primary diagnostic test. And it's I would be reluctant to predict how long it's going to take to get sequencing technology accurate enough and cheap enough. I think it's a few years away. Preliminary research and clinical studies show exome sequencing can be useful clinically, as I said, as a secondary test to chromosome microarray. And whole genome sequencing is going to continue to improve its sensitivity and accuracy to detect CNVs, and will become cost-effective. It's only a matter of your guess versus others, how long this is going to take. I think it's going to take at least a couple of years, maybe more like 5 years. And at that point, we might transition to first-tier testing by whole genome or exome sequencing, and postnatal, prenatal, including non-invasive testing and cancer diagnostics, although many of those things will still require follow-up testing to determine mechanism, inheritance, and recurrence counseling by standard cytogenetics and FISH analysis. So I'll just close by thanking my collaborators at Emory and now at Geisinger, and particularly Crystalise Martin, who spoke yesterday afternoon, who's been a longtime collaborator in all of the research activities I've spoken to you about today. So thank you very much for your attention.
Okay, first of all, I want to thank you for a great presentation. Really appreciated it. As a QA/QC type person,